So thank you very much. So I'm from Pfizer, and I have a co-presenter, Jacob, who will be presenting the technical stuff in the middle of the talk. So we're looking at adverse events, and can we get those from different places? And we have lots of different places we can acquire the adverse events from. We can look at PubMed, we can look at FAERS, look at pharmacovigilance data, we can look at social media and other places, even internal studies. But the one place we're really looking right now is focused on the drug labels from the FDA and later from the EMA. So this is our source of insight on what's going to make a good drug. So drug labels, I thought, were these things on the side of boxes, which I never read in the first place, except to find how many tablets I should take. But that was until last year, when I realized that these drug labels are 30 to 100-page booklets that sit inside these medical things that you need a magnifying glass to read. But they have a ton of information on them. And so they have all sorts of things about the indications, about exactly how you use it, the dosage, who's it for, contraindications, warnings, precautions, adverse events, interactions, and all sorts of other stuff that I had never appreciated before because of the fine print. So if we're looking at drug labels for trying to understand what the adverse events are, there's only a few sections that we really need to look at. And so there's just these four: the warnings and precautions, the adverse reactions, the use in specific populations, such as pregnancy, lactation, and so on, and the non-clinical toxicology groups. So we've taken these 30 to 100-page labels that can focus on these four sections mostly, but that doesn't make the problem that much easier. Where do we get these drug labels from? Well, we don't have to go through the medicine cabinet. We can actually go through the FDA website called the Daily Med. And they have a wonderful site you can search for all the stuff, where you can download the entire set of all the drug labels and all the information, either as a PDF or an XML file. So this is a great source for lots of information. And so the specific project goal that we set out to do is, can we identify the adverse events associated in each drug label? So that was the major one. But in addition to that, we wanted to know, well, what dose and what administration route was associated with that adverse event. And this is true not only of the humans, but also the animal models, which are part of the preclinical, and the species was the last aspect. So we thought this would be difficult because there's been competitions in the past. There's many competitions that have tried to extract this over the years. But the technologies of NLP have gotten so much better in the last few years with the deep learning, the BERT models, and so on, that we thought we'd have a go and see if we could do it internally as a separate project. And so we initially focused on just the FDA labels, and we're going to focus later on the EMA labels, but not in this talk. So the first step is extracting the XML documents, parsing those things. If you haven't seen one for a while, it kind of looks like this, which is not readable at all at the back, but it has a bunch of information, a bunch of tags, kind of like HTML. And so all the information is in there, but you have to dig for it. And even a pretty formatted table is, again, a pile of XML, so you have to go parse those things. But there's lots of tools available for it. And we elected for the XML rather than the PDF parsing, even though the EMA only uses the PDF. So the next thing was, can we identify the clinical adverse events? All right, this is our main problem, but is that hard or easy problem? So here's 3 examples. Weight gain has occurred with drug X. So weight gain is all together. It's in the same sentence as our drug. This one's pretty easy, because we can just look in a dictionary for weight gain as an adverse event, and we're pretty well golden. The next one, patients treated with drug X experienced increases in both ALT and AST. All right, so I have to know what ALT and AST are, that they are bad things, or they're liver enzymes, but it's the increases in them that's the bad thing. And is it increases in ALT, an increase in AST, and the modifier corresponds to both of those things, right? Because it has the word both, but it's English, so it's a bit complicated. So it's, again, a medium difficulty. The last one, there is no evidence of increased prevalence of Stevens-Johnson syndrome related to drug X. So not only do we have to look for the positive cases, but we have to look at and understand the negative cases, which, again, is more difficult. They're separated by different words. We have to identify and understand the structure of the sentence in order to be able to figure it out, plus know what prevalence is, as opposed to just the Stevens-Johnson syndrome. And so there are many challenges for that. And so we brought in our secret weapon, Jacob, who's going to take over here.
Alrighty, thank you, Peter. So I'm going to start going over how we actually approach this problem. Specifically, we started with using what's known as MedDRA, and it is a 5-level hierarchy system specifically for identifying and labeling adverse events within human clinical trials. And it has a hierarchy of where we start with a organ system class, and this could be something such as a gastrointestinal disorder. We drop down to a higher level group term where we have gastrointestinal signs, drop down again to a high level term, nausea symptoms, again to just nausea, and finally we can get down to some of the adverse events that we're looking for in the lowest level terms, such as feeling queasy. And now this dictionary has around 130,000 different terms at these different levels and over 80,000 terms at the lowest level. And what we did is for the at least clinical adverse events is we built a dictionary using these and started to find patterns within these sections in these FDA drug labels. So here's a successful adverse mining with MedDRA. So we built these patterns to identify adverse events using MedDRA, and we can start to see them highlighted within the text specifically. In each of these cases, we have this event identified and tagged, and when we go back through and want to pull these out later on, since they're tagged, we can easily do that. What we start to see, though, is within MedDRA, we have laboratory measurements. However, the phrasing within MedDRA is very limited. So in the cases of these, we have AST increased, AST decreased, AST flare-up, low, high. And these are the only ways that this is mentioned within MedDRA itself. When we start to have different phrasings, such as AST elevated to or AST went down, MedDRA is no longer able to capture these. So how do we go about fixing this? What we did is created additional dictionaries, such as the modifiers dictionary we created, which included increasing, high, elevated, terms like this that start to quantify how these different laboratory measurements are being changed over the course of clinical trials. So in this case, we can see, pulled straight from a FDA drug label, we have an event tagged, we have the percents that these occurred at, as well as a hemoglobin measurement, as well as an increase modifier. So in general, these lab measurements are limited within MedDRA as well. So when looking at, for example, iron binding capacity, that's not actually within MedDRA and would be completely missed if you're relying on that method alone. So finding MedDRA phrases is insufficient. Again, these AST increase, decrease, low, and high. When we start to run into examples such as experience increases in both AST and ALT, we aren't going to be able to capture that both these increased because the increase in the AST are not directly next to each other and the phrasing is different. Other examples would include such as increased levels of AST were sometimes found, as well as this no increase in prevalence. So we have to look for those negation terms as well. So what we use to fix this problem is Spacey, specifically for parsing up sentences, taking different parts, and then looking at the word dependencies within these sentences. So in this case, we have a sentence, the following laboratory experiences were reported, sophonelia and hemoglobin increase. And if we dial in upon this one part of this hemoglobin increase, we can see that the increase is directly related to the hemoglobin and modifying it, and we can look for those dependencies based off of what we tag within the sentence and then put these adverse events together. So an output in this case would just be the increase in hemoglobin. Moving to a different project goal, specifically we wanted to look at the route of administration, such as an oral tablet, as well as the dosages that these adverse events occurred in. What we used is a pre-trained model specifically for named entity recognition. In this case, all this information was stored within the dosage administration sections. So as you can see, we have our drug, tolvaptan, and its form, tablets, automatically recognized within the text. And if we look down further, we can see that we have several different strengths and dosages, frequencies, that we can link together in much the same way we did the laboratory measurements and the modifiers. So in this case, we would have a form of the drug as labeled as a tablet, our 15 milligrams linked to a once daily, the 30 milligrams links to a once daily, as well as the 60 milligrams linked to a once daily, to get those complete dosage information for this drug alone. Moving from the clinical adverse events, we started to look at the preclinical adverse events, which are animals. And what we found was that MedDRA terms are for humans only, and we start to run into a lot of problems when we try to apply that to animal terms. So in the cases of we have a shortened tail, that's not going to ever occur in any sort of clinical adverse events, a decreased reproduction rate as well, or any sort of mention of an exoskeleton. That's not going to be covered within MedDRA and are not going to be captured within those terms. So what we did is we started with building new dictionaries to try to handle this. And what we started with was VEDRA, which is from the EMA, and is like MedDRA, but for veterinary medicine. What we found was that helped cover a lot of different cases. However, that did not cover everything. So we started to look out and branch out even further into mammalian ontologies, mesh, as well as building our own species list that we were interested in. So rabbits, mice, dogs, cats. But what we found is that there was still a lack of standardization of preclinical terms, and there was hundreds of different ways to get the same message across. So our solution to this problem was to use named entity recognition. So it is a use of natural language processing to classify word categories from context. So it doesn't matter how the word is being said or phrased, it's still going to recognize that as an adverse event due to the encodings that it uses. So this relies often on handcrafted training set of data to learn from the contexts. So what we did is we had to go through individual sentences pulled from these clinical documents, tag them individually with our events, and then feed it back into our algorithm to get these better results. What this did was it allows for discovery and classification of previously unseen adverse events that are not in the dictionaries. So if in the case we had an adverse event that was never mentioned or in any sort of VEDRA, we could start to identify those, which we've never been able to do if we were relying on a dictionary method or pattern matching alone. Another unique problem that this solved was that it was able to distinguish between what was a blind study and an adverse event, such as a night blind, or night blindness, I should say. And so we started to get good results from this. So when we look at for training, we want to see the adverse events in different positions within the sentence. So in the first case, we could see cause an increase of some sort of adverse event at the end of the sentence. Or we could also see something sometimes caused a decrease of adverse event, or adverse event were observed in some patients. And finally, we could have it in the middle of a sentence, such as some patients showed signs of adverse event after long-term use. And in each of these cases, our model, our named entity recognition model, is going to be learning the distinct characteristics of how it's being used within those sentences and use that context to identify it. So what we did was we custom trained our named entity recognition model for preclinical text. So we took a large number of preclinical text samples, labeled them using our various different methods, and then fed it in to try to get an idea to train our model and then use that to validate it as well. What we did is we focused on 3 categories of text, which would be adverse events. So this could be the bruising, the increased reduction rate, or shortening of tails. Other ones we looked at were modifiers. So again, this is the increase, decrease. And finally, we looked at the medical or laboratory tests as well. So think blood tests or any sort of biological markers. And what we did is that we leveraged that clinical training with MedDRA for training these preclinical models. So what we ended up with is that we achieved 80% accuracy on all the preclinical text. So roughly 80% of the events that we were hoping to tag, we would tag. And we also identified events from contexts that were, 1, not part of the training data, so this is in our validation set, and also not found in any of the ontologies. This is, again, finding these new adverse events that would never have been labeled if we just relied on dictionary methods. And finally, it worked with the different word orders, so we solved the problem of having to have specific wordings that would cause these hits. So in the end, this is a sentence pulled from preclinical text, and we can see that we'll have rabbits, in this case highlighted, and then our multiple animal events highlighted. And in this case, we can see that more complicated events such as embryo-fetal mortality are captured, as well as other ones such as skeletal malformations, which are not easy to do with just phrasing alone, or with patterns alone. We also see that in this case, we have our dosages labeled as well. So we can capture everything pretty well using this method and combining it with the pre-trained model for dosages already. So I'm going to pass this back to Peter, and he'll talk about comparing it to a standard.
Thank you, Jacob. So as you can see, what's been done is that we've been able to extract all the adverse events, mostly for humans, and doing that pretty successfully. That was with the MedDRA. And the second part was, can we get stuff for the animals where we don't really have a very good dictionary? And that's been a tough challenge, but with the named entity recognition and other approaches, we can do a pretty good job. We're getting about 80% in many cases, and we're still kind of a work in progress at that stage. So I think we have a few more levels to go. And so we're about to celebrate and give ourselves a self-high five, I suppose, but are we done? The answer is no. We really need to compare the results to a standard. It's not good enough that we can pull out the right text and say, this is the adverse event. We really need to normalize it against something else. And so just to motivate that, we have drug X has all these different adverse events. It's 4 different ones, and drug Y is 3. So these are completely different, or are they the same? We'd really like to say, these are all the same thing. And the adverse event occurs both in drug X and drug Y. And so we need to map these things to some other criteria that we have or don't have. So this is the new challenge. MedDRA has, for example, some issues. There's no word for fever, which is how I would normally to characterize a high temperature. They have the word pyrexia, which just doesn't come to mind. Closest thing is Pyrex jugs. So they also use British spellings, and so we're not going to have fetal heart in the FDA records as well. So more challenges. And lastly, there's an issue of what level do we actually map back to if we're using MedDRA? Because we could map out to the adverse event level, or is it something below that? It's neurobehavioral changes, or more specifically, long-term neurobehavioral changes. And if we get some of the words right, we get on some level, but if we miss some words, is it correct or not? Again, we really need to map back to the ontology, which has the multiple levels, therefore we can assess exactly where we are. And this has been the new challenge. So we're approaching it using an embedding approach, and this one's word2vec. This is a fairly famous approach from Google about 2013, 2014. They've trained a model on lots of different texts, and they look at that particular word in the context and say, what are the words around it? And so if they do that for lots and lots of texts, but typically medical texts or biomedical texts, then they can assert a particular number associated with every single word. And so dizziness gets a massive vector of all these different numbers. Migraine gets a different vector. And this is really important because what it's done is it's quantified meaning, quantified the context and said, well, this set of vectors, this set of numbers in this vector describes exactly what this means. We don't know what the numbers mean, but we can compare the similarity or dissimilarity between those vectors and come up with an assertion whether or not this is really the same thing or something different. And then we have to take those different parts and glue them back together again. So this is working reasonably well at this point. We still have some little ways to go. The challenge is that it's word2vec and not phrase2vec. We can train up a new version, but it's difficult. And so given all the training that's already available in these dictionaries, we can find the femur, we can find length, we can find reduced, but we got to put those things together somehow. That's not always as trivial as it sounds. And is heart and rate together the same thing as independently? It's really one concept. So we're still working with some of those issues and different ways of representing things, but it does find us the right synonyms and the right level of granularity in many cases. And so here's an example of a section from a mouse and section from a human. And I won't read it to you, but all the text in blue we've been able to identify automatically and pulled it into a nice table, which is what we're looking for at the end of the day. Some level of granularity that says, yeah, this is the drug name, here's the event, the adverse event. We have a certain dose that we've recognized it either from the particular section or from an earlier section in the document. And it's orally administered, and it's one's from a mouse, one's from a human. And so this is working quite well as shown here. but we're not quite done. We're still working on it. We've extracted most of the adverse events. We can use the MedDRA plus the dosage plus different modifiers like increased and decreased, put those together and do a very good job for the human adverse events. We're using SPACY to take those different word groups and then map them together so we can identify how those different entities relate to each other so we can associate that increased AST and so on. The preclinical is still the challenge, and I'd love to hear any ideas from the audience on how to handle the preclinical standardization. We have that the MedDRA, which is for veterinary stuff, but it's really not a standard for adverse events of animals. It's got all sorts of stuff in there. It's got different body parts, different species, and all sorts of other things that really aren't exactly what we're looking for. We're looking really for something that's a list of all the adverse events associated with animals that we can eventually relate to humans in many cases. We don't have that. So we're in the process of exploring how to build that dictionary because I think the field needs one. But that will help us map things back to some standard. And so this last part is the normalization. How do we map the words that we identify and correctly extract and put them back to either MedDRA or some other future standard as we have for animals? And that's all I have. So thank you very much for your attention. Happy to take any questions.